and we'll start where we finished off. So I think it was stem cells. So I included stem cell research in this chapter because you are manipulating DNA in many cases. So stem cells are undifferentiated cells found in many parts of the body. They're used to regenerate damaged or dying tissue, to replace those cells. If you try to grow them in cell culture, they do fine. They grow like a normal cell, not like a cancer cell, which piles up on top of each other, but stem cells will grow and get a what's called confluence. They just grow right up next to each other, then they stop growing. But if you grow stem cells, which is an immortal cell line, in cell culture, they don't do anything. They just behave like any other, like a bacteria or something. They don't differentiate or anything. It's when you take a stem cell and you put it into an existing matrix, then it has all the growth factors and all the different signaling systems that tell it what to do, which could either be differentiate into a skin cell, if that's where it is, or differentiate into some other kind of cell. So they respond to signals in their environment. So if you take any cell from the human body or an animal, it can't be a stem cell. But embryonic means from an embryo, and bone marrow stem cells are pluripotent, which means that they can reproduce endlessly and differentiate into pretty much any type of human tissue. So then we have induced pluripotent stem cells. They can be made from some types of differentiated cells by reprogramming them. And the reason why we have to deal with that is because some countries ban the use of embryonic stem cells because they think that the embryo is a person. And if that embryo could get a lawyer, they would sue you. So anyway, it's been a very important topic. How do we do stem cell research without using embryonic stem cells? And so here's one thing that was tried, and this was really interesting because this was done in a very prestigious journal, which means that it's a very important topic. And they claim to subject blood cells to near-death conditions and not reprogram into stem cells. But then the paper had to be retracted. Nobody could repeat the results. One of the researchers committed suicide. Half the other ones were fired. Apparently they just faked it. And stem cell therapies have skyrocketed. There's a clinic in every state. They claim this, they claim that. And the business get around legal regulations by claiming that the treatments are not a drug. So it's a very hot topic, very controversial. So here's a couple of examples using stem cell research. So this guy founded his stem cell institute, his own company. He injected 15 elderly people with stem cells and showed that it was safe. And then he took 30 elderly people and showed that there was improved lung function and stamina to walking and all kind of stuff, just because they were treated with stem cells. It would make them younger again. Human embryonic stem cells were implanted into mouse beta pancreatic cells and then transplanted into diabetic mice. So the mice began producing insulin and were cured in 10 days. So many of the stem cells are created by what's called somatic cell nuclear transfer. A somatic cell is a cell that, because of limitation of telomerase activity, can only divide maybe 20, 30, 40 times, then it dies off. So they transfer the nucleus from a female's egg and put it into the nucleus of the foreign donor cell. So in this case, they claim to not be using an embryo. It's from the female's egg. See, they're doing this just to get around the concept that an embryo is a person and has rights. Then it grows this mass of cells, and those are stem cells. So there's been scattered medical successes using stem cell therapies. Here's one. In 2016, they took embryonic stem cells and put them into patients with Stargardt's macular dystrophy, and they showed marked improvement in the degeneration of the macula, was stabilized in several others. There seemed to be no adverse effect. And here's something interesting too. So this happened in Hawaii. So this couple wanted a baby, but both parents were carriers of thalassemia. So what that means is that there is a good chance that their offspring would have this disease. This is the disease where they can't make enough red blood cells. And so neither one of them had the disease, but they were carriers. They had one copy each. So if a baby were to receive 2 bad copies, then they would be suffering from the disease. So anyway, they took stem cells from the mother's hip bone, that's bone marrow, and used that to basically create an embryo. And then the baby was born free of disease. So about 8 years ago, they found out that women can produce egg cells throughout their life. Changes in hormones is sometimes what causes the shutdown for stem cells to differentiate into egg cells. So with hormone therapy, at least in animals, they're able to get stem cells from these egg cells. And this is kind of important because there's only 2 white rhinos left. Both of them are female. And so they're trying to do a similar type of thing. They froze male sperm before the last male died. So they're trying to keep that species alive. Here's a couple other things. I won't go into every single thing. Stem cells can be used to grow new organs. So they're being used to create blood vessels. So they make a biodegradable mesh in the shape of a blood vessel. You seed it with human vascular cells that came from cadavers. Now, some of these are going to be stem cells. So basically, you're using stem cells to create blood vessels. And speaking about cadavers, so my good friend and colleague, maybe it was about 10 years ago, was interested in stem cells. A person had just passed away and donated his body to the university. And he went down there and immediately got brain cells from this cadaver. He was able to isolate stem cells from the person's brain after he had been dead for about 2 days. And he kept them growing in his lab for, 6 months or a year or something like that. So speaking of that, skin cells were induced into pluripotent stem cells and then turned into what's called mini brains. So here's some pictures of these mini brains. They're stained with different protein binding fluorescent stains right here. And so why would they do this? because now they can study these brain cells to see how they respond to different environmental factors, how they respond to drugs. They're not active brains, obviously, but they're brain cells in a brain environment, and they can actually be used to study all kinds of different things. So you've probably heard about this, growing tissues in different organisms. So when you have a mixture of cells from different organisms in the same tissue, that's called a chimera. Okay. So here's an interesting thing. So the cornea transplants were done using corneas from cadavers. About a third of them would fail. But if they could get corneal cells grown and transplanted to repair corneas, then one donated cornea could create treatment for hundreds of patients. So a couple years ago, they did this with 11 patients. They took fresh corneal cells, magnetized them with nanoparticles. That means that they use basically a modified gene gun, and they shot the corneal cells into the person's eyes, and then they could use the magnetic fields to adjust where the cells were. Later on, the nanoparticles just come off and come out of the body, and they're hoping that that's going to cause the regeneration of these people's corneas. So a couple other things in there. They're talking about making artificial wombs, all kinds of different stuff. Then cloning. Yeah, I'm sure you've heard about cloning. So in 1996, they made Dolly the sheep. And everybody thought, oh, wow, that's so amazing. That was doing somatic cell nuclear transfer. But it took 277 attempts. So what they found is that the nucleus, how do you say? The reason why it's not easy to do is because in higher organisms, the DNA is just so wound tightly that it's hard to actually do it. So recently, some Chinese researchers produced 2 monkeys that are identical twins using cloning. So there's a way around this. Anyway, I just wanted to give you guys some ideas of what kind of stuff is in the news. Also in the news is GMOs. So I've been following GMOs for a long time. I think I was one of the original ecologists. Before I ever even went to, before I went to a 4-year college, I was doing a lot of diving, working on environmental impact statements and in the marine environment. So that was like maybe about 73 to 76. And I did that to basically work my way through college. So even before that, I was just like, why are you guys wasting so much plastic? And now I see all these people who are against GMOs, and they tie it all into being, in favor of the ecology and saving the planet. But, it's the exact opposite. So about 4 years ago, pretty much every living Nobel Prize winner signed this letter that went to Greenpeace asking them to stop the GMO bashing and stop getting in the way of getting nutritious food to those who need it. And to me, I just find it so bigoted where these people are like, well, you should go to down-to-earth foods and you're getting all this organic stuff. Yeah, down-to-earth foods is expensive. What about the people who can't afford it? So Greenpeace led the fight against what's called golden rice. Golden rice had the potential to provide vitamin A to the poorest people in Africa and Southeast Asia. They suffer from vitamin A deficiency, subsequent diseases, and the GMO crops have been studied intensely and found to be safe. In fact, most GMOs are not transgenic, meaning that either an existing gene was altered or a gene from a close relative was added. So in other words, you take a gene from one variety of wheat and put it into another one. So you can design wheat plants using wheat genes that are tolerant to drought or salt or things like that. With CRISPR technology, the accuracy and everything else is becoming much better than before. And the FDA only is going to regulate transgenic foods. That means you take a gene from, say, a giraffe and put it into, a monkey or something like that, trying to get a monkey with a long neck. That would be transgenic and not even practical anyway. So the first genetically modified organism was a bacteria, 1973. Then the first animal was since 1974, the first plant in 1983. The first commercialized GMO food was the Flavor Saver tomato. And all they did was make it ripen more slowly so it would be okay to withstand shipping with time. Then the first commercial GMO animal happened just recently. That's the Aqua Advantage salmon. So it grows fast, it gets large because they put extra copies of the gene that codes for growth factor into the salmon. So it turns out that plants are readily changed by genetic engineering because all the plant cells are what's called totipotent, meaning that every cell can give rise to become any other cell in a plant. So a single cell has the potential to become a whole plant. So if you take a single plant cell and you modify it the way you want it, that can grow into an entire plant. So anyway, humans have been manipulating genes for at least 6,000 years. Plants, animals, whatever. We do it by hybridization. We do it by selection. We do it by controlled mating. That's how you get all kinds of different dog breeds, right? Even though they are all the same species. So the Green Revolution avoided a predicted famine. Famine was predicted after World War II because of the population explosion. And the reason why there wasn't a famine is essentially because of fossil fuels. So fossil fuels were allowing the production of fertilizers, the spreading of the fertilizers, planting of fields that weren't plantable before, the shipment of foods from one continent to another. This is the reason why there wasn't a huge starvation. But now the population has increased dramatically again, and we're facing the need for another green revolution. So here's kind of this paragraph right here kind of shows you what ecologists are concerned about. And I remember judging the science fair probably 8 years ago. And these two girls had gone around and were measuring the DNA in papayas. So they had these farmers that thought they were growing natural, non-GMO papayas, but in fact, most of their papayas were GMO because of cross-pollination. And so, yeah, I mean, there has to be precautions taken. So GMOs have sparked worldwide concern, leading to riots, crop vandalism, I should say, laws being passed, but yet decades of research have failed to find any conclusive evidence that there's anything detrimental to eating GMO foods. So here's the reason why we need GMO. Here's the total death toll from great famines. You see that they happen quite often. And here's where we had our green revolution over here. And here's the population growth. You can see we're headed towards a problem over here. So here's some examples of non-transgenic GMOs. And it actually includes foods that are put into much of our other processed foods. Okay. Pretty much anything with corn in it, anything with olive oils and all this other kind of stuff. Okay. So if we look at some of the concerns from the anti-GMO people, they say GMO is a radical technology. Well, not really, especially with CRISPR, where we're so accurate now. Haven't been studied long enough? Yeah, I think we've been studying it quite a bit. Here's one that I keep hearing. Farmers can't replant the GMO seeds. So they talked about these Terminator seeds. Well, Terminator seeds were never approved. They never made it out of the patents office in 1990s. That's 30 years ago. And if you're a farm, you barely have enough time to get seeds from wherever and plant them and grow them and harvest them and take care of them. You don't have time to create another complete business, which is to take some of those and get the seeds necessary for the next year. You simply buy them from someone else. So, but that's something that's still being used all the time against GMO. Do they cause allergies? No, those are tested all the time. Big agriculture funds GMO research. Well, they make it sound like it's been a big secret thing. We personally have several graduate students in ag science, and their graduate research is being funded by what was then Monsanto, you know, the big evil Monsanto. And I kept asking them, does Monsanto even know what research you're doing? And they said, no, they don't care what we're doing. They just want to invest in research in agriculture. So GMOs require greater use of pesticides and herbicides. That's not true either. So here's where that comes from. So Bt is a protein that comes from this bacteria. It's toxic to insects. So organic farmers that did not want to use pesticides and herbicides were spreading Bt protein on their plants to kill the insects, thinking that, okay, this is natural, it's not a pesticide. Well, that is the protein that was put into certain plants so that now the insect would bite the plant and then die. So instead of spreading it around on the farm, it's in the plant itself. It is the protein, and you know what happens to proteins when you eat it. Then glyphosate is an herbicide used to kill weeds that crowd out desired crops. It's one of the most environmentally friendly and mild herbicides, 25 times less toxic than caffeine. And resistant crops allow the use of glyphosate without killing both the weed and the crop. So the use of glyphosate was dramatically increased, but alternative herbicides were way worse. So despite all the lawsuits, the FDA does not recognize glyphosate as a carcinogen. So you may have heard of people being paid millions, if not billions of dollars because they claimed that glyphosate gave them cancer. Well, there's a little bit of science would ever agree to that. But in a court of law, all you have to do is convince a jury of your peers. So the lawyers find peers that don't know anything about science. So GMOs create super insects and super weeds. That's possible. If you use anything too much, you will kill off all the ones that are resistant and then leave room in the ecology for the resistant organisms to survive and then take over. So farmers still have to practice sensible farming. Same thing with antibiotic resistance, right? So GMOs harm beneficial insects. Well, there has been a problem with the monarch butterfly. Talked about it. There's nothing hidden about it. It's true. But then again, you got to go back to this. Farmers have to practice sensible farming. Yes, cross-pollination can happen. So just the other day, I went to Tropical Agriculture's seed department, and I was asking them for some rainbow papaya seeds. The rainbow papaya seeds are resistant to the ring spot virus. And they said, oh, you have to watch a 1-hour video and then sign some paper that you know how to handle GMOs. And I said, okay, fine. But what I'm trying to get across there is that, yeah, they take huge precautions with this stuff. It's not like we just throw it anywhere we want. So RNA interference technology is another way to improve crops. It was used to downregulate omega-6 fatty acid desaturase in soybeans. What does that mean? So you make an RNA that binds to the messenger RNA that would normally code for this fatty acid desaturase. So that means that that enzyme is knocked down and you get lower levels, or I should say you get higher levels of monounsaturated fatty acids. So you improve the oil. So here's another example of genetic engineering, a gene drive. So what a gene drive does is it spreads a trait through a population rapidly and to every individual. So you genetically modify a mosquito. You introduce a gene that's designed to also cut the other gene in the new offspring. So when the wild gene is repaired, it gets converted into the new gene because that gene is used as the repair template. Okay? So here's the introduced gene, right? That comes from the male or the female. Here's the from the other one. So you damage this, right? And then it goes through a repair process and it uses this to cut and damage that. So it has to be replaced. And what's it replaced with? Itself. So now this organism, when it goes through sexual reproduction, both of the offspring will get the gene drive. See how that works? So what can you do to wipe out a mosquito population? Well, you could do a couple of different things. You could make them so they don't mate. You could make them so they don't live long. But what you want to do is make it so they don't die off before they mingle in with the rest of the population. You want to mingle them in with the rest of the population, Then when they start mating with that population, the gene propagates through that population. So if enough modified mosquitoes were released, a new version of mosquito would dominate in the wild after only a few generations, which is basically a few months, right? Altering 1% of the wild population could eradicate mosquito-borne diseases within a year. So here's some of the things that they've been trying. And then, if they're ready to do that, they can do it. They haven't done it very many places except in the lab because they're afraid of what it might do. So another company is trying something different. They produce a bacteria strain that infects mosquitoes and prevents their eggs from hatching. So what they're going to do potentially is release 20 million infected male mosquitoes in California to see if it would dramatically reduce mosquito populations. So when we get to the chapter on viruses, you'll see how many viruses are actually transmitted by mosquitoes. Okay, so here's another one using bacteria. So besides that, Hawaii lost most of its endemic bird species to avian malaria. So yeah, if we got rid of the mosquitoes in Hawaii, yeah, we'd be able to save much of our existing bird species. So here's a couple novelties. So last year, they made an E. coli with a completely reconstructed genome. It took 5 years, but what they did is they synthesized small DNA fragments, inserted them into existing bacteria, then cut out some other ones. They kept going until there was no more, original DNA. Okay? So they still grow. They have a little bit different shape, grow a little bit slower, but it was pretty important accomplishment. And then in 2014, they had announced that they made a completely synthetic chromosome that was functional in baker's yeast. So baker's yeast is used in cooking, of course, and stuff like that. Many of strains of that is used to make beer and things like that. So what they want to do is do the rest of the chromosomes, and then they will have a completely bioengineered yeast. And the idea is that they know the exact sequence. They could use CRISPR, for example. Well, let's take out this gene and replace it with another one to get the exact strain of yeast that we want. Pretty interesting. And here's an extra supercalifragilistic super GMO bacteria, E. coli. What they did is they invented a new base pair. So not GCAT, but XY. They invented this. And then now you have not 4 to the 3 because there's 3 codons. 3 bases in a codon, right? That's what we have with our existing DNA. But you got 2 more bases, which would give you possibly 216 different amino acids. It's like, what the heck? So it's possible that they could create proteins with more than 20 different amino acids. They can invent their own amino acids. So that's kind of the idea. So it's not totally functional right now, but pretty interesting idea that they're actually working on. Huh? I remember when I was a grad student working in the chemistry department, some of the senior professors that I knew, they're like, wow, you biochemists don't even know this and don't even know that. And I'm like, yeah, you guys think water is a contaminant. One day we were grading an exam and they just kind of got me mad. So I started talking about, yeah, if you really want to do stereochemical correct organic synthesis, you better start using enzymes. And that's the whole thing about this, right? Creating these superorganisms that we can control, the enzymes that they produce are what's really important to us. So anybody have any questions so far? So we get all this genetic information. Who owns the information? You think you do, but you don't really. There's some more expensive companies that will keep track and protect your data. But the Golden State Killer raped over 50 women, killed at least 13 people. You probably heard about how they caught him. So a distant relative had done a genealogy with GED match, and so they used a combination of DNA found at crime scenes, DNA from this distant relative. That narrowed it down into 100 suspects that could have been the Golden State Killer. But anyway, so if you look at where the crimes were committed and where each of these 100 people were, there was really only one person that could have been responsible. And they didn't get a warrant to get his DNA. They just found stuff that he had thrown away, discarded items. And you see that now on TV shows all the time. They won't give the DNA, so they get it off a cup or a soda can or something like that the suspect had discarded. So some people still see this as a breach of privacy, but the website is free to the public. So 17 other suspects have been arrested for such crimes. So anyway, we give off millions of skin cells every day. We're spreading our DNA everywhere. So if you go to Starbucks and you leave your DNA on a cup, throw it in the trash can and go away, is that really private information still? It was left in a public area. Interesting legal concept there. On the other hand, if everybody just gave their DNA to these databases, we'd have a much better database. It would be much more relevant in terms of racial equality and all that kind of stuff as well. So what about genealogy? So humans share 99% sequences with Neanderthals, about the same amount with chimps, less with baboons, less even more with bananas, but still 44.1% the DNA is the same. Any two people share greater than 99.9% of their DNA. So anyway, Ancestry and Geno and all those kind of things can pretty much give you this kind of information. Okay, so here's a person. This is where their genealogy came from. And how do they know that? Well, it has to do with mostly mitochondrial DNA, because that's easier to track, mutates more slowly, has a better or more constant rate of differentiation. So here's another one, 23andMe. This is what, I forget where that came from. Anyway, you get the idea. People have done it. And so what's interesting is that the DNA in most modern humans can be traced back to an exodus in Africa that happened about 40 to 80,000 years ago. Occurred over a period of time. And here's some interesting information about that. And I've had discussions with this about Hawaiian ancestry. And yeah, it's pretty interesting how they respond to that. So my really good friend is, you know, a cultural practitioner and all this kind of stuff. And he had his DNA tested, but it's not really good enough to say, well, oh, he's 50% Hawaiian or something like that. It's not good enough to do that. So paleogenetics, the oldest DNA analysis is from a horse that lived more than 500,000 years ago. But DNA degrades faster than proteins. So now they're trying to do it also with proteins, right? So using mass spectroscopy of proteins, they've been able to look at proteins from, say, a rhinoceros from, you know, 1.8 million years ago. It's crazy. Okay. What about hominids? So in 2014, they sequenced the entire mitochondrial genome from a 2,000-something-year-old hominid. They've done it for 40,000-year-old Neanderthals, and anyway, they do have limited information, very limited information. But this is where all this information comes from about, you know, what really happened with humans and everything else, and how are we related to each other from different continents. So here's kind of the basic story that we know so far. So Homo sapiens and Neanderthals both originated in Africa about 500,000 years ago. The Neanderthals migrated to Europe, while the ones that remained developed into Homo sapiens. And then these populations developed, well, they were destined to turn into separate species. Then about 100,000 years ago, migrating Homo sapiens went into Europe from Africa and began interbreeding with the Neanderthals. So people think of Neanderthals as being stupid and ape-like and whatever. Well, you know what? Apes are not even that stupid. And so more modern days, we think that they were intellectually and culturally advanced, both groups. So some people are proposing that they were simply subspecies, and they want to give the names Homo sapiens sapiens and Homo sapiens neanderthalensis, okay, just to show that they were so closely related. So what happened to the Neanderthals? Well, some people say, oh, we killed them off. Well, maybe we did, but it depends, because it wasn't one wave that moved from Africa into Europe. It was a constant migration. So the Neanderthals probably didn't live in large groups. They were spread out through the environment. And then when a few of the first African-based Homo sapiens started to come, they started to interact. But then when more of the Homo sapiens started to come, they probably started killing them off. Humans are really good at that, aren't they? One of their favorite things. So they found a 90,000-year-old fossil, 90,000-year-old fossil that was half Neanderthal and half Denisovan, which is another type of Neanderthal. Came from the Denisovan caves, okay? But this is where they're really thinking right now. So Homo erectus may have given rise to this Denisovan Neanderthal, which then turned into Homo sapiens. Then some migrated into Europe, and then later on those mated with these Homo sapiens and turned into Homo sapiens. But we're all the same species. Anyway. So now they're looking at methylation patterns to see how the physiology of something like a Neanderthal could be different when Homo sapiens have so much of the data, same sequences, right? So that's a very important thing. So methylation patterns are different between now and between a long time ago. So that's another point that they're trying to look at. So this HOXA gene was kind of important because if you suppress the expression of the HOXA genes, you get heavier bones and you start to get looking more and more like a Neanderthal. So you ever hear of the Tasmanian tiger? Okay. So there was a movie, actually. It was called The Last Tasmanian Tiger. It was not a bad movie. It was about this guy who was set out to hunt it because they thought it had, curative properties. But then he ended up trying to protect it. But anyway, it's a real thing, and it's actually a marsupial. And look how wide his mouth opens. It's a marsupial that ate other marsupials, kangaroos, wallabies, all that kind of stuff. So that large mouth, that large opening probably allowed it to grab onto, those larger organisms, though, to catch it, right? So anyway, this is a picture of the last one known in captivity. So some people are like, well, let's bring it back. So here's the list of organisms that they're thinking about bringing back. Okay. So there's a couple ways to do it. Here's one. They took DNA from a frozen ibex, extinct, implanted it into a goat egg cell, and the baby ibex was born to a goat. It's called a clone. It lived only about 10 minutes, but that proved that it is possible. You ever see a Tasmanian devil? These things are just nasty. They're not that big, thank God, but their bite is just extremely infectious. They're carnivores and they're just mean. So they're trying to say, well, wow, we have the Tasmanian devil. We can use that to maybe create the new Tasmanian tiger. But anyway, I'm like, well, maybe that guy should be extinct. Maybe this guy should be extinct. Another thing they're thinking about is, well, how do we just use normal hybridization to reverse engineer the sequence of an extinct species? And I see some of my friends and relatives do that, actually. They're into pit bulls, right? So they'll find a pit bull that's small, and then there's another pit bull that's small. Then they take the two pit bulls and breed them, hoping that the pit bulls will be small. So it's the same kind of thing. It's like breeding to get the features that you're looking for. But whether that would really happen, I don't know. So anyway, that's about it. Yeah. Look, you two were made for each other. Pretty funny. I think it's funny. Does anybody have any questions about this chapter? No. Well, let's start the next chapter. Okay, come on. What's going on here? Here it is. So I made two chapters out of the chapter that we just finished, one on cancer and one on viruses. And so one on viruses I'm still working on. I just finished this one yesterday. So cancer is a genetic disease. There has to be damage to a gene, either acquired, inherited, or something that happened to it, from the environment. And tumors are simply fast-growing cells. They grow about the same rate as a fetus. They cause the symptoms when they apply pressure to nerves and organs and start to shut down their normal function. Okay, and so they do not respect the presence of other cells. Like I was saying about stem cells, when you grow them in culture, they grow to what's called confluence. They grow right up against each other, and then they stop multiplying. Well, cancer cells just ignore them and keep multiplying. So what are some characteristics? Well, they're basically pretending like they're not part of a multicellular organism. Okay, it's all about me now. I'm not part of this organism. I'm just going to multiply, divide, raise a family, do whatever I want. So most normal cells wait for an external message to to divide. Cancer cells can counterfeit their own growth message. So as a tumor grows, it puts pressure on surrounding cells. Those cells then issue chemical messages to stop growing, and the malignant cells ignore that. When normal cells are damaged, they self-destruct by apoptosis. Cancer cells do not do that. Tumors need nutrients and oxygen to survive, and they often cause nearby blood vessels to form new branches to feed the tumor. It's called angiogenesis, generate new blood vessels. Normal cells, senescent cells, divide about 70 times until they start losing too much terminal DNA and they start to die off. Cancer cells have extremely high telomerase levels, so they can just divide indefinitely, become immortal. So the first 5 could describe a benign tumor, which may or may not be life-threatening. The cancer cells can also metastasize, which means that some cells will break away from the original tumor, maybe push their way between the cells that make up a capillary, get into the capillary, go off and colonize another tissue. In terms of ecology, which I think we do need to start looking at cancer cells in terms of ecology and Darwinian features and stuff. They're really an invasive species. They try to take over every niche as possible. So there's a couple of theories about how cancer develops. One is the oncogene tumor suppressor hypothesis. And so you have mutations all the time. If a mutation happens in a tumor suppressor gene, you lose that growth suppressing protein. So then the cells can simply grow unrestrained. And so another thing that can happen is a mutation in an oncogene, this is one that produces a cancerous protein, can become hyperactive, producing oncoproteins that cause the cell to just grow wild. Okay. So of course, epigenetics can be incorporated into this whole theory, but here's kind of the idea. If I damage this oncogene right here, an oncoprotein can come out here and simply turn on genes, cause the cell to proliferate. Here's a tumor suppressor gene. Damage in here will stop the production of a tumor suppressor, so it's no longer able to tell an oncogene to stop being active. So both of these are most likely regulatory genes, especially this one right here. So another modification of that is that a mutation results in damage to the machinery involved in DNA replication. And then repair leads to thousands of random mutations. Okay. So eventually the cancer genes we see, observe, become mutated. So you're attacking the genes that are involved in DNA replication, messing up that system. And the early instability process suggests that mutation in a few genes is required for cell division results in corruption of the chromosomal structure. So now when cell division happens, you have chromosomal abnormalities. Okay. So anyway, there's a little bit more to that theory, but that's the basic idea. And so the all-aneuploidy hypothesis suggests that the first step towards cancer results from an abnormal number of chromosomes in a cell after cell division. So in this hypothesis, they're saying that happened by accident and it created a property that was passed down to further cells that are metastatic, I should say, cancerous. Here, a mutation occurred in the genes that are required for cell division, also giving rise eventually to aneuploidy, which is a disruption of the number of chromosomes. Here, the mutation is in DNA and repair genes. Here, mutation is in an oncogene or a tumor suppressor gene. So it's most likely that all four of these are involved. So what is an oncogene? It's simply a gene related to cancer. And so here's an interesting problem. So P53 stands for protein 53 because it was a protein with a molar mass of about 53,000, is one of 36 known tumor suppressor genes. Okay. Its products, P53, inhibit cell proliferation. So when a cell has experienced damage due to several types of stress, P53 binds to the duplex DNA and prevents cell division. So that gives time for the cell to either repair itself or go through apoptosis. And so the mutations in the gene for P53 are seen in about 40% of tumor types. So when they were able to increase P53 in a mouse cancer model, it was found that they did not get cancer. Anyway, but then is that a way to prevent cancer in people? No, because those mice developed all kinds of other problems and they died 20% earlier than control mice. So the more cells you have in an animal, the greater the possibility of cancer. So that's observed in large dogs versus small dogs, also in humans. But there's some large animals like elephants and whales, and in some other smaller ones as well, that have a low incidence of cancer. So that's called Peto's paradox. So how can an elephant, which probably gets cancer, I think I read that somewhere, maybe 2 or 3 times less than humans do, but yet they have so many more cells. How is that possible? Well, they looked at about 53 animals, and most of them had a single active leukemia inhibiting factor. That's a gene. And then they found that elephants and a few other species had multiple copies, with elephants having between 7 and 11 copies. And most of the time, genes with multiple copies, which are called pseudogenes, only one of them is active. In the elephant, they found at least 2 were active. So when they took elephant connective tissue and grew it in cell culture, cancer-inducing stress activated this gene and the cancer cells were killed. When they suppressed that gene, the elephant cells formed tumors. They also found that the activation of this gene requires another gene to function, and there's 20 copies of that gene. So what that gene codes for, well, is a protein that forces cells into apoptosis. So blocking production of the T53 protein also blocked the activity of the other gene and forced the cells into apoptosis. So we know a lot. There's been hundreds and hundreds of experiments that are very important to us understanding cancer, but yet we can't really explain all the different kinds of cancers we get. There's not a single principle. So what I'm going to do is just go through some of the recent findings. So for example, oncogenes in some tumors are actually less active than in normal tissues. It's like, what? So after 30 years, the oncogene tumor suppressor gene hypothesis failed. Doesn't make sense. So CT scans, nearly 20% of all tumors detected in CT scans are so slow growing that they would never cause problems to the patient. So in other words, you're not going to live long enough for many of these tumors to actually cause you any harm. So knowing that, we have to go back to the data, and it looks at the number of patients whose lives were saved by CT scans. You got to revise that, because in at least 20% of those tumors that were found by CT, you're pretending like you saved their life when you didn't really save their life. Then we know about primary and secondary tumors, and what we really should be doing is the treatment should be focused on the driver genes responsible for formation of both primary and secondary tumors. So a primary tumor and a secondary tumor are probably genetically different. They're going to behave differently. So treatment for one is not necessarily going to be the treatment for the other. But what can you do to prevent a primary tumor from creating a secondary tumor? And then they found cancer stem cells. So here's what they did. Here's the experiment. They created a mouse a knockout mouse with no immune system. And then they took a tumor and they took it apart into different kinds of cells. What they found when they took most of the cells and implanted into the mouse without the immune system, it never formed a tumor. However, certain cells, which were called cancer stem cells, would cause a tumor. So this is really important for us to understand, because if we take this tumor and we treat it with chemotherapy, for example, we kill off how many cells? But all it takes is for one cancer stem cell to survive, and that tumor is going to regrow. The idea is that cancer stem cell is the true progenitor of the tumor. And it's changed how all these other cells react, but these other cells are simply doing whatever this stem cell is telling it what to do. Okay? So anyway, I just drew this. I didn't know, I was trying to figure it out myself. So if you take a normal cell, once it differentiates, those are normal cells, they die and replace normally. If you get DNA damage outside the bone, you create tumor cells. Those could be long-lived. But if you get DNA inside the bone, and then it goes through the process, that is what could be giving rise to cancer stem cells. Okay.
So would a bone marrow transplant be the answer to a stem cell tumor, or is that still unknown?
It probably could. So in a bone marrow transplant, what they do is they basically kill your immune system. They kill the cells in your bone marrow and then replace them. So yes, that could be done. Yeah, theoretically.
That sounds horrible, though.
It sure does. Yeah. And you may be on immunosuppressive drugs forever. And the other thing is that because stem cells are so long-lived, they basically start accumulating all this genetic damage. So anyway, one recent discovery was that in childhood onset kidney cancer, some cells seem to be stuck in an immature fetal state. So the cells that created the tumor acted like stem cells, and so kept proliferating instead of differentiating, which goes along with that theory of cancer stem cells pretty well. So epigenetics could be a problem. Say you have a tumor suppressing gene, and then for some reason, epigenetics methylates it and turns it off. Now that's a problem for you. And we talked about telomerase. So telomerase has been linked to shorter telomeres, which is linked to aging, infectious diseases, heart attack, drinking, smoking, and stress. And I find this bizarre, but studies with children have shown that telomere erosion is increased by stress, by them seeing violence or whatever, right? So stress is a very important thing. I was reading an article one time about mid-sized cats, predatory animals in Africa. So, for example, the cheetah. The cheetah could easily be killed by another leopard or tiger or a lion or something like that. But it has to live by killing other smaller prey. So the cheetah has to make a decision. when it chases down, say, a gazelle or something like that, it may use up so much of its energy to do that, that it's just lying there, not able to defend itself from some other predator. Matter of fact, I've seen videos on National Geographic type things where the cheetah runs down the animal, but it took so much out of the cheetah is just like laying there, huffing and puffing, not able to kill the animal. The animal gets loose and runs away. During that time, the cheetah is susceptible to being killed by any other thing. So it's a stressful lifestyle compared to, say, the apex level predator, and their lifespans are much shorter. So it's an example of me where stress basically causes shortening of lifespan, probably because of telomerase activity. So they found DNA enhancers. So pancreatic cancer cells have been shown to carry short DNA sequences called enhancers that activate certain genes that lead to metastasis. And when it comes to personalized cancer treatments, or what's known as pharmacogenomics, there's been a couple of different movements over there. So this is one. So this is an anti-cancer drug that inhibits this particular enzyme and that particular gene. So what it is, here's the drug. It's a competitive inhibitor to ATP for kinases. So here's a kinase, and here is the drug bound to the kinase. So it's in clinical trials, trying to treat anaplastic large cell lymphoma, neuroblastoma, and some other solid tumors. So what they're doing is they're making fusion proteins. They're doing all kind of stuff like this. And the whole idea is this. This is a drug that works on some people, on not other people. So how do you know which people it's going to work on? What kind of cancer? That's why there's the Cancer Genome Atlas. And it basically individualizes tumors so that, oh, you get this kind of tumor, well, this drug should work on it, this drug might not work on it. So it's personalized medicine. There's another company that looks at 70 genes associated with the risk of recurrence of breast cancer. There's all kinds of stuff like this going on. Then another new thing are checkpoint inhibitors. Well, not exactly new. Type of immunotherapy. So cancer cells were able to avoid detection by the immune system by releasing what's called checkpoint proteins. What those do is suppress immune system genes. So checkpoint proteins are a naturally occurring things. So sometimes cells will protect themselves against overactive T cells by putting checkpoint proteins out there. So the T cell comes along, okay, am I going to destroy you? No, you have this protein, you're okay. So the cancer cells have produced a whole bunch of these checkpoint proteins. Okay. So here's one, PD-L1. So some cancer cells have upregulated the gene for PD-L1 to protect themselves from attack by these T cells. It causes the T cell to undergo apoptosis. So originally this is probably a mechanism to keep the immune system in check. There's another one, and here's another one. So what are checkpoint inhibitors. Inhibitors are antibodies or enzyme inhibitors that target checkpoint proteins. Okay? So if you can inhibit the tumor from producing checkpoint proteins, then the immune system can then attack that tumor. That's the idea. So we have at least 5 checkpoint inhibitors, which are antibodies for the most part. A combination of 2 of them used on advanced melanoma patients increased the 2-year survival rate by significant amounts. Same with lung cancer. But there's harsh side effects. Harsh side effects. Anyway, they're very expensive, harsh side effects. Here's some of the ones right here. I mean, you've seen some of them advertised on TV. I've seen Opdivo. I've seen Keytruda advertised on TV. Yeah. But there's no checkpoint inhibitors for the most common cancers. Okay? These are solid cancers. They block the immune system. It's harder to get treatments to a solid tumor than to a less solid tumor. And yeah, but then again, there's always surgery with subsequent treatment. But yeah, it seems like all tumors are going to require multiple kinds of treatment. And then recently they found in some cancers there's a subset of the tumor cells that are resistant to checkpoint inhibitors. So here's another one as an immunotherapy. It's the CAR T. So they call it a living drug. So they take the patient's stem cells, genetically modify them, and then put them back into the patient. So what it does is you genetically modify the patient's own T cells with a gene for a receptor protein that allows the T cells to bind to an antigen. So first of all, to be effective, the cancer cells have to have this CD19 antigen on their surface. So a normal T cell is not going to recognize that, but they created a gene that codes for a brand new receptor protein. So that becomes synthesized on the surface of the T cell. And when it binds to the CD19 receptor, it can attack that cancer cell and kill it. So where does the CAR T come from? Chimeric antigen receptor on the T cells, so it's called CAR T. So mice with human melanoma tumors were cured when the engineered T cells were able to locate and destroy the melanomas. So they basically cured a girl who was close to death from acute lymphoblastic leukemia. So leukemia is a blood disease, it's not a solid tumor, right? Out of 63 patients treated with another study with this same one, 11 died, but 52 went into remission, which is pretty significant. But the harsh side effects are a problem. Okay. Not only that, it's extremely expensive. So right now it's considered a last resort treatment, but at least it's something that people can look forward to. You know, don't get your hopes down. But if CAR T fails, then well, that's kind of it, huh? So another thing that they're trying to do is you come down with a tumor, and instead of doing something like CAR T, what they're doing is they're taking the tumor and comparing the patient's T cells and looking for a group of T cells that will then attack that particular tumor. So it's like the tumor from the patient and the T cells from the patient, and they're looking at which T cells, if any, can attack that tumor. If they find one, they'll take that T cell and reproduce it, grow it outside the patient, and then inject it back into the patient. So there's been some success with that. Yeah. So there's a rare mutation causing an autoimmune disease. And so the T cells from those children were corrected with CRISPR. So that's still in the works. The original results were published, but I haven't seen any follow-up on it. So let's see. Here's another one that was done. Okay. So modifying the patient's own T cells, putting them back, and see what the T cells can do. So here's another strategy. So IDO stands for indoleamine 2,3-dioxygenase, also known as tryptophan dioxygenase. It's in the pathway to synthesize NAD. So I put the pathway down here. Here's nicotinic acid. You basically get NAD, right? So here's the enzyme over here. It's the first enzyme in taking tryptophan and using it to create NAD. Okay, so this is close to nicotinamide. That's the reason why. This is nicotinamide adenine dinucleotide. So if you were to block this enzyme, you don't create any of these other products, right? So this enzyme has been shown to be involved in how cancer cells avoid the immune response. So they found that the expression of this enzyme was high in several types of tumors. And so they spent like $1 billion. Pharmaceutical companies spent about $1 billion trying to bring inhibitors to this enzyme into clinical trials. You see how this would be so easy? All you have to do is, what does this enzyme bind to? Oh, tryptophan. So to inhibit it, you just need to take tryptophan and start making analogs of it. Put a methyl group here, put an ethyl group there. If you come up with an IDO inhibitor, it's simple. That's why they spent so much money on it. So anyway, it's a very unstable enzyme. It's hard to learn about how it works and everything. So it's been really difficult to make inhibitors to it, just because you can't just easily isolate it in the test tube and let's test what inhibits it. So they originally thought it would be easy, turned out to be not so easy. So I also found out that inhibitors, let's see, where am I now? Well, they can make antibodies to IDO, and that may also be able to cause tumors to stop growing or to shrink. Anyway. And then similarly, they found tropomyosin receptor kinase. This is in the brain. It was found to trigger tumor growth in the brain. So they developed this drug right here to inhibit the gene for this enzyme. I don't know whether there's been any success with it or not. Then the amino acid asparagine was shown to be necessary for breast cancer to spread in mice. So metastasis is what they're looking at. So they're thinking that a diet low in asparagine, or then treated with asparaginase, in other words, take the food, treat it with asparaginase, would decrease the number of malignant tumors outside of the breast tissue, and it was. Okay? So it didn't stop the primary tumors, but it stopped the metastasis of the primary tumors that would normally go off and form secondary, tertiary tumors. So how does that work? Nobody knows. So the Zika virus was shown to kill neuroblastoma cells in cell culture studies. So neuroblastoma is a super deadly brain disease. I've seen people die from it very quickly. So that's just something they're looking at. Okay. So the Zika virus obviously causes brain problems in babies and fetuses, but not in adults, right? So here you're going to treat an adult with this. So then looking at mitochondrial proteins, they found that this anti-cancer drug, this is in clinical trials for glioma and some other cancers, known to stop proliferation and kill tumors in cell culture. What does it do? They found that it activates a mitochondrial protease with that name. It cuts up misfolded proteins in mitochondria. Okay. So that's a pretty new thing. And then interference RNA. They were hoping that this would be helpful in fighting cancer. It looks like it's probably not. Okay. So this is something that they'd hoped for. So interference RNA knocks out the messenger RNA coming from certain genes, right? But it hasn't been successful. Matter of fact, drugs from simplex DNA, which would be used to bind messenger RNA, and RNA interference, doing the same, have been used to knock out cancer genes, but they're not very effective. You can't get them to go to anywhere in the body you want, and they're also degraded fast. So just recently, they came up with this, spherical nucleic acids. It can get into every part of the body, including the brain, before they're attacked by the immune system or by nucleases. So they got single-stranded DNA designed. Its sequence is going to attack certain messenger RNAs, right? It's attached to a nanoparticle that's made up of a liposome. There's an anchor protein that anchors the DNA to the liposome. And the DNA strands kind of point out like a sea urchin spine. So it allows multiple cell surface receptors to bind to it. So when they made this, it was like, how is this drug going everywhere in the body? And they found out that it basically, all these different spikes sticking out can bind to multiple receptors on a cell, causing the cell to bring it in phagocytosis. So the single-stranded DNA then binds the intended messenger RNA. And right now, no treatment has been approved, but there's been some cases right here where it shows that it could be an excellent way to make RNA interference that doesn't work using DNA. So I just noticed that we're out of time. Does anybody have any questions before we stop for the day? And again, with this chapter, what I want you to think about are the basics. There's millions of examples of the basics, right? But concentrate on the basics. You're never going to be expected to know, what does Yescarta do or something like that. You know what I mean? So any questions about anything?
No questions, but I did find it really interesting. Like, I need to pay more attention to the new journals and everything being published on this.
Yeah. Yeah, and if you only watch the journals, there's too much complexity there. There's too many things going on. So what you need to do is look at review articles.
Oh, I see.
Otherwise, you'll never be able to keep up. You know what I mean?
Yeah, and a lot of times the language too is a lot too complicated for me to comprehend.
Yeah, but after this course, I mean, that's part of what you learn from this course, right? Is, telomerase. What is that? Well, now you know.
Exactly.
So that's the idea. Okay, well, tomorrow's lecture, I think, will have to be at 1:30, so I'll send you a notice of that. You guys have a great day, and I'll finish doing your exams right now.
Thank you. You have a good day.
Thank you. Take care. Bye.
Bye.